Hello students. Today we're going to talk about the lymphatic system and the immune responses and different types of resistance to disease mechanisms that our body has to protect us against pathogens and other infectious agents. Disease resistance occurs in our body in two principal ways. We have something called non-specific resistance or innate immunity. That just means that we are born with certain defense mechanisms that have the general capability of eradicating pathogens from the body in a non-specific manner. And we're going to talk more about what that means in a few slides ahead. And then pretty much everybody knows about immunity. We have an immune response. So when people think about immunity, they talk about really what we call adaptive immunity. And adaptive immunity is the type of immunity that we acquire in order to activate our immune system cells called lymphocytes that can eradicate specific pathogens. So sometimes adaptive immunity, or what we call immunity, is referred to as specific resistance to disease. So although pathogens and infectious agents are eradicated from the body both by non-specific and specific mechanisms, they work independently of one another. So we're going to talk about these two types of specific and non-specific resistance to disease mechanisms and how they work in this chapter. First, we have to talk about the lymphatic system as a whole and what type of organs and structures are associated with the lymphatic system. And then we'll talk about the basic functions before we get into disease resistance. So the lymphatic system is comprised of really three things. It's comprised of a fluid which we call lymphatic fluid, which flows in lymphatic vessels throughout our body. You see all the little green vessels in this picture. And specific organs, which are specialized reticular connective tissue organs. That means they have a lot of reticular connective tissue in it, which you remember that term from A&P 1. So the organs in the lymphatic system and tissue in there that's going to bring about some of the functions of the lymphatic system include red bone marrow. Here they show the proximal epiphysis of the femur, where red bone marrow is located, and all the epiphyses of long bones. There are specific little organ structures along the lengths of lymphatic vessels, which are referred to as lymph nodes. We have the single largest mass of lymphatic tissue in the body called the spleen. And then we have the thymus gland. So we have lymph, the fluid lymph, flowing through lymphatic vessels, which goes through lymph nodes with organs like the thymus gland, the spleen, and red bone marrow. So we need to look at what those are and what the roles of these organs are in the lymphatic system. So all throughout this PowerPoint, like the other ones that you have been reviewing, have these links that I would like for you to review. You have to be connected to the internet to view the animation, so make sure you are. And just review those. It's going to help you out a lot, especially in this system. This system has some information in it that may be a little difficult, so you might want to watch all of the animations. As far as the functions of the lymphatic system are concerned, there are three. The lymphatic system is involved in what we call fluid recovery, which is when there's an excess of fluid in a tissue, or the interstitial fluid flows back into a small lymphatic capillary. And so the lymphatic system is going to help collect fluid that drains out of the blood capillaries that infiltrates tissues. And some of that fluid, about 3 liters of fluid a day, is recaptured into the lymphatic system. And as the fluid is being collected from the interstitial fluid and going into lymphatic capillaries and then into lymphatic vessels, it's called lymph at that point. The lymphatic system also is the system that transports dietary fats from our intestines. So the only nutrient in your diet that is not absorbed directly into the cardiovascular system, into a blood vessel in the intestine, are fats. Sugars, carbohydrates, proteins with amino acids, vitamins, minerals, and water go into the cardiovascular system, whereas fats are absorbed into the lymphatic system first. And we're going to see where lymph actually drains the fluid, where the lymphatic system actually drains lymph and those dietary fats in a minute. Ultimately, the lymph fluid is drained back to the cardiovascular system, so we're going to see that in a minute. And then obviously the last thing we're going to talk about is the immune responses and how the immune responses are carried out via the lymphocytes, the T lymphocytes and the B lymphocytes in the body. Lymphatic vessels ultimately begin as a blind or closed-end capillary, which is different from a blood capillary, which those are all open-end. But there's a closed-end capillary, very small lymphatic capillary, which collects fluid from the tissues that are leaking out of blood capillaries, along with some leaked out proteins. And that fluid then drains from a lymphatic capillary into larger lymphatic vessels, which resemble cardiovascular veins because their walls are thin and they have many valves in them, which is going to allow or help for the flow of lymph through the lymphatic system. So here's a diagram showing a capillary bed over here. Here's the arterial feed of a capillary bed. Here's the capillaries, and then we have the venual drain from a capillary bed. And here's all the body cells in the middle, along with the cardiovascular capillaries or blood capillaries are also lymphatic capillaries. So lymphatic capillary is blind-ended or closed-ended, as you can see here. And in the upper picture up here, we can see that some of that fluid that ultimately leaks out of the blood capillary re-enters the lymphatic capillary. So this is part of that fluid recovery and recovery of leaked out proteins and whatnot that I was mentioning before. So those lymphatic vessels ultimately pass through the body, and lymph is flowing through the vessel that passes through lymph nodes. Lymph nodes are a type of lymphatic organ that we're going to talk about in a minute with its structure and its function. But as the lymph flows through these lymph nodes and the lymphatic vessels start to join together, they form larger lymphatic vessels, which are called lymphatic trunks or lymph trunks. And the trunks in the body include the lumbar, intestinal, bronchomediastinal, subclavian, and the jugular trunks. Now these trunks are named for the area of the body with which they are collecting lymph from. So obviously the lumbar trunks are in the lumbar region. The intestinal trunks are draining or receiving lymph from the lymphatic vessels from the intestines and the mesentery. And then the bronchomediastinal are receiving lymph from the vessels in the thoracic cavity, so forth and so on. The lymphatic trunks join together to drain the lymph into two major lymphatic ducts. There's a left lymphatic duct, which is referred to as the thoracic duct, and a right lymphatic duct. So let's look at that for a minute in this picture. I'm not going to put this picture on the test, but you should look at it so you can see where the lymph is flowing. So down here at the bottom, you can see this would be the thoracic cavity in the picture. This is the abdominal cavity. In the abdominal cavity, lymph is being drained from the lower appendages up from your legs and in your hip region up to this region. The lymph is draining to this central location called the cisterna chyli, and we have right and left lumbar trunks and an intestinal trunk, which is draining lymph all into the cisterna chyli. Lymph then flows from the cisterna chyli back up through all of these vessels and the trunks in the thoracic cavity, and ultimately drain into a right lymphatic duct over here. This is the right side of the neck. and the left lymphatic duct, which is also called the thoracic duct. And the lymph drains via the right and left lymphatic ducts into the area of the junction of the jugular and the subclavian veins. So ultimately, all the lymph that's flowing through our body, carrying also our dietary fats, drains back into the cardiovascular system up here at the junction of the jugular and the subclavian veins. You can see a more detailed picture of it here as the lymph is coming up from the right side of the body and then the left side over here. Now, this is just a schematic, not a very detailed drawing of the flow of lymph and the association with the cardiovascular system. I want you to review that. It talks about the flow of interstitial fluid into lymphatic capillaries, as I was mentioning before, and the various vessels, trunks, and ducts that it goes through, ultimately draining into the subclavian veins. So this area right here on this picture represents the area where the jugular and the subclavian veins meet, and thus the right and left lymphatic duct will drain lymph back into our blood in the cardiovascular system at this spot. So ultimately, all the fluids in our body originate from filtration of plasma somewhere from the blood, and so is lymph. Lymph, the fluid lymph, actually is a collection of drained or filtered water and other solutes from plasma, and ultimately that fluid in the body returns back to the cardiovascular system at some point. In the case of lymph, it occurs in our neck at the junction of the jugular and the subclavian veins. So this picture shows a representation of a blood capillary bed here with a lymphatic capillary, either in the pulmonary circuit or the systemic circuit, and fluid will leak out in both areas, and we have lymphatic vessels, capillaries, and vessels that will collect that fluid, then pass the fluid, lymph, through lymph nodes that basically stud along the length of our lymphatic vessels, and the lymph nodes are going to be like little filters where we can ultimately have our immune system cells monitor the fluid for any infectious or pathogen, infectious agent or pathogen, and then we can rid the body of that through our specific resistance to disease mechanisms that we'll talk about. So let's talk about the primary and secondary lymphatic organs in the body. So all of the organs I was mentioning before, part of the lymphatic system, can be separated into primary or secondary lymphatic organs. The primary lymphatic organs consist of two things: red bone marrow and the thymus gland. The primary lymphatic organs are the organs where immune system cells, the T cells and B cells, what we call T and B lymphocytes, are either produced and or become immunocompetent. So we know that all blood cells are produced in red bone marrow. So the white blood cells, the T and B lymphocytes, which are white blood cells being produced in red bone marrow, would make red bone marrow a primary lymphatic organ. The thymus gland is considered a primary lymphatic organ because that is the place where immature T lymphocytes, called pre-T cells, migrate from red bone marrow. Once they're produced, they migrate there in the blood, and that's where they become immunocompetent. So this term immunocompetency refers to the ability of the T and B lymphocytes to carry out an appropriate immune response to eradicate a pathogen or infectious agent from the body. So when the B cells and the T cells are produced in the red bone marrow, they're not immunocompetent right when they're made. They have to basically mature before they gain that ability to fight off infectious agents. And we're going to talk about that process. So the thymus gland is located in the thoracic cavity, just superior to your heart in the mediastinal area, and just deep to the sternum. So you can see here in this picture, the sternum, it has been removed, and the thymus gland lies pretty much on top of where the superior portion of the heart would be. And the thymus gland is one of those primary lymphatic organs. The structure of the thymus gland is includes, I should say, capsule, connective tissue capsule that surrounds the entire gland, with little extensions from the capsule that dive into the gland, which are called trabeculae. And those trabeculae partially lobulates the gland into little sections, which we call thymic lobules. The thymic lobules consist of an outer cortex region where immature T cells will infiltrate that area and start to become tested. And we'll talk about this testing in a minute, but they start to become tested to see if they can gain the ability to fight off infectious agents without hurting our own cells in our body. So part of the maturation process is occurring here. And then they migrate more towards what we call the medulla, and this is where we find the more developed, immunocompetent, mature T cells will then enter back into circulation. Secondary lymphatic organs and tissues are the tissues and organs, I should say, that actually carry out the immune responses, where basically mature, immunocompetent B lymphocytes and T lymphocytes are located. The secondary lymphatic organs include the lymph nodes in the body, stationed along lymphatic vessels, the spleen, which we'll see in a second, and then something called lymphatic nodules. So let's take a look at some of these organs. Here is a lymph node. Lymph nodes are stationed along the length of lymphatic vessels, and in which case lymph will be delivered to the lymph node via several different afferent lymphatic vessels that enter the convex side of a lymph node. You see how it's rounded on this side. So the lymph will flow through the vessels into the lymph node in this direction through an outer cortex. So there's an outer region of the organ called the cortex. The most outer portion of that cortex is called the outer cortex. And in that outer cortex are specific types of lymphatic follicles or nodules that I'll mention in a second. And then we have an inner cortex, which pretty much includes the majority of all of the follicle, not just the top of it. So the outer cortex, just underneath the capsule, with what we call the subcapsular sinus. So the fluid flows through there, enters the inner cortex through the lymphatic nodules. We then have the lymph flow through what we call the medulla region, the medullary region, which is in the middle, and then it flows out via two efferent lymphatic vessels. So the lymph always flows in afferent vessel through the outer and inner cortex, through the medullary region, and then out through the efferent lymphatic vessel. If there's any pathogens or infectious agents in that lymph, the infectious agent and the pathogen will be introduced to our immune system cells as they're located in these various regions. So if you notice up here in the outer cortex, there are B cells, cells called follicular dendritic cells, which we'll talk about, and macrophages, which you guys are probably familiar with already. In the middle of a lymphatic nodule, there are mainly B lymphocytes in an inactivated state. The middle of this lymphatic nodule, or follicle as it's called, is called a germinal center. The germinal center is called that because that's typically the area where B lymphocytes begin to become activated against a particular pathogen. And once it's activated against a particular pathogen, it becomes a plasma cell, which ultimately produces antibodies against that particular pathogen. The inner cortex contains T cells and dendritic cells predominantly, and those particular cells, the T cells, can also become activated inside of a lymph node if a pathogen is being introduced to them. We're going to talk about this activation process in a minute, as it is a very important process dealing with specific resistance to disease. In the medulla, in the middle of the lymph node, we have B cells, activated B cells called plasma cells, and macrophages. And We're going to talk about what all of these cells do to help eradicate pathogens in a minute. So just know the flow of lymph through the lymph node. You can see the little caption down here. Just make sure you're familiar with that. And what areas of the lymph node, the different types of cells, where they're located. Here's a lymph node, the histology of a lymph node. I'm not going to put the picture on the test, but we can see the capsule up here. We can see the trabecula, which is an extension of the capsule, which helps lobulate the organ. We have an outer and inner portion of the lobule, whatnot. And so these are the areas that I am referring to when I talk about outer cortex, the inner cortex, and the medullary region. So you could just review this. I'm not going to make you identify these regions, but we are going to get into the physiology of how these cells are acting and where they're acting to become activated against particular pathogens. The spleen is the largest mass, the largest organ in the lymphatic system. It's located in the upper left quadrant of your abdominal cavity. It's just lateral to your stomach, and the kidney. There's a left lateral depression called the renal impression, where our kidney indents into the spleen. The spleen is an encapsulated organ. It is a secondary lymphatic organ, meaning we may carry out immune responses there, but the B cells and T cells are not produced there, and they don't undergo maturation and become immunocompetent in the spleen. So it's considered a secondary lymphatic organ. Now, the tissue in the spleen is separated into two main groups, something called white pulp that you see here. You see all these, the darker purple area loaded down with different types of white blood cells that I'll mention in a minute. And then the other area called red pulp. There are scattered white blood cells there, but many red cells, and that's why we call it red pulp. Also, platelets are stored in this region. In the very middle of this cylinder-looking structure called white pulp is what we call a central artery as well. The central arteries perforate the areas which we call white pulp. So, the spleen contains red pulp, white pulp, and red pulp. The reticular fibers, collagen fibers, fibroblasts, which are loaded down in the spleen, is part of what we call the stroma. The stroma of any organ is the structural framework of the organ. And so the stroma of the spleen includes the connective tissue capsule that surrounds the spleen, along with the extensions from the capsule called trabeculae. Reticular fibers help support the structure of the spleen, and fibroblasts producing all the connective tissue fibers forming the stroma. The functional area of an organ is called the parenchyma. The parenchyma of the spleen includes white pulp, where many different types of white blood cells would be located, like our B and T lymphocytes, which carry out immune responses, and many macrophages in there as well. And then red pulp is the area where we might have some scattered white cells there, but it's predominantly red cells and platelets in that region. Red pulp is an area where the macrophages will remove defective red blood cells out of circulation that we covered in chapter 19. Remember, red blood cells only live for about 120 days or so. So the macrophages in the red pulp region are phagocytizing those dead or dying red blood cells, and it's pulling them out of circulation. The red pulp also is the area where our body can store up to a third of its platelet supply. In case we need more platelets to enter circulation, they can be removed from the spleen to enter circulation. Now, in the adult, the spleen is not involved in blood cell production, but it is in the fetus during fetal development when the baby is developing in utero. But the spleen loses that ability to produce red blood cells and white blood cells and platelets after we're born. Lymphatic nodules are part of what we call our secondary lymphatic tissues. They're often referred to, since they're not encapsulated, they're often referred to as mucus associated lymphatic tissues, or MALT. And these little collections of lymphocytes without a capsule around them are scattered through what is referred to as the lamina or propria of our mucous membranes. The mucous membranes that line your gastrointestinal tract, your urinary tract, your reproductive tract, and your respiratory airways are all lined by these lymphatic nodules containing T and B lymphocytes. So if the membrane itself is compromised, whether it's through an ulceration, let's say in your stomach, or some abrasion in the esophagus, or any other damage to the epithelial membrane, if there's a pathogen trying to enter through that open area, those pathogens will come in contact with our T cells and B cells in the lymphatic nodule, in which case they'll become activated against them and eradicate them from our body, basically producing an immune response. So lymphatic nodules are pretty important in helping activate B and T cells throughout mucous membranes in our body to aid in immune responses. You can view this animation, Introduction to Disease Resistance, pretty good animation, talks about the two types of resistance mechanisms that we're going to be getting into. So let's start that. Innate immunity. Innate immunity basically involves all of the things that we are born with that help prevent either infection, or that is getting the infection, and or eradicating the infection from the body. So this is often referred to as what we call non-specific resistance to disease. And the pathogens and or their toxins, which can cause problems in our body and make us sick, can be eradicated in a non-specific manner. So we have two basic types of defense, which we call our first and second line defenses, which are non-specific. Skin and mucous membranes is our first line of defense, which obviously we're born with that. We have our skin and our mucous membranes developed in our body. The skin and the mucous membranes around the body are a physical barrier for the entry of a pathogen into the body. So they actually form the barrier by which a pathogen is prevented from causing infection. It works most of the time, but it's not 100% effective. Especially if you have a cut or an abrasion or a burn in your skin, you open up that particular physical barrier and you produce what's called a portal of entry. A portal of entry is basically a hole in one of these physical barriers that allows the entry of a pathogen. So if our physical barriers are uncompromised, they're not cut or broken, they do a pretty good job at preventing infectious agents from entering our body. We then also have internal defenses, which include several different things. Chemical defenses that include the production from your sebaceous glands you learned about in AMP1, sebum. That's the oil. It's acidic, helps prevent bacterial infection. Lysozyme is an enzyme that's found in our tears, our saliva, and our sweat. This enzyme helps break down the cell walls of bacteria and thus destroying them. And then we have gastric juice, which is acidic, hydrochloric acid. So if we are consuming foods or fluids that have pathogens in them, luckily for us, the majority of them are destroyed in our stomach. There are some that are not, but most of them are. They are destroyed by that acidic environment. So our physical barriers that I was referring to before are lumped into also what we call mechanical defenses. So that includes your skin and your mucous membranes. So our fluids in our body, like tears, saliva, mucus, cells that have cilia on them, your epiglottis protecting your trachea, the opening of your trachea, the flow of urine through the urinary system helps dislodge bacteria if they're in there, other pathogens, and it'll flow out of the body. Going to the bathroom gets rid of defecating, gets rid of pathogens, and even vomiting. When we get sick, we vomit. That's our body's reflex to help get rid of pathogens that might be trying to infect our body. So these are our first and second line defenses, which are inherently in our body already. So they are what we call innate defenses, or non-specific resistance mechanisms to disease. Including in our internal defenses, we have our various types of proteins produced by cells in the body, like white blood cells and other cells, that can destroy microbes. They're called just generically antimicrobial proteins. There's many different types. Some cells in our body that can basically eat up bacteria or other microbes shown in this picture, like phagocytes, can help gobble up or eradicate pathogens through what we call phagocytosis. We have natural killer cells, which are more like T lymphocytes than other cells in the body, but they work in a non-specific resistance manner, which means they don't care what type of cell is entering the body. If it looks different from our own cells, they'll try and go and kill it. So they're called natural killer cells. And then I'm sure you heard about inflammation. We'll talk about inflammation in a minute, and fever in the body. Both of these are part of our innate immune response, non-specific resistance to disease responses that aid in eradicating pathogens from the body. So during phagocytosis, there are 5 basic steps. If you don't remember this, you should review it. It's not too terribly difficult. Chemotaxis is the ability of our white blood cells and phagocytes, like macrophages, to follow a chemical trail where they can hunt down microbes that are trying to infect our body somewhere. They then go through something called adherence, where they start to wrap their plasma membrane around the pathogen. Then the pathogen is ingested into what we call a phagosome. The membrane of the phagosome was the membrane of the plasma membrane that wrapped around the pathogen. And that phagosome is fused with a lysosome, the cellular stomach, in which case, even though they don't say it here, this is called a phagolysosome, and the digestive enzymes and acids in the lysosome are dumped all over the pathogen, and you can see during the progression of the process here that the pathogen is being destroyed, and then ultimately it kills it, which is the final step, killing. They don't show, but this vesicle can then exocytose out the dead parts of the pathogens out of the body. So that's one way we can get rid of infectious agents, through phagocytosis. Now, the inflammatory response is a natural response to any type of tissue trauma or other stress or damage in our body. When inflammation is not controlled is when it's bad. A hypersensitivity reaction. Too much inflammation can cause our own tissues to be destroyed, which we don't want. So it's sort of like an allergy attack, is a hypersensitive inflammatory response, and you feel real bad, so you have to go take Benadryl to block histamine to help subside those symptoms of the allergy attack. But inflammation is not all bad. We need inflammation to help repair our tissues and eradicate pathogens. So inflammation occurs via immigration of white blood cells into tissues. They then begin to eradicate the pathogens in several ways via phagocytosis, the release of several chemicals. In the case of neutrophils, we learned about in chapter 19, like lysozyme and defensins, and strong oxidants like hydrogen peroxide and bleach helps eradicate, in this case, bacteria that are infiltrating the wound site. In order for the cells to immigrate efficiently, part of inflammation includes vasodilation of the vessel and an increase in permeability of the vessel, so more fluid leaks out, more blood enters this area, and the cells are allowed to go into the tissue more easily. Since we are increasing blood flow to the area and we're increasing fluid leakage from the vessel into the tissue area where inflammation occurs will turn red, it will get hot, and it becomes swollen. Those are 3 of the 4 signs of inflammation. So the area where we have inflammation turns red because more blood goes there, and blood is red. It swells up because more fluid is leaking out of the blood vessel, and it is hot because blood carries heat with it. Now, the fourth sign of inflammation is pain, and pain comes about because of several different things. It can come about from the tissue trauma itself, from the release of various compounds, either from the microbes and or our own cells, just tissue damage in general, and irritate our nerves and whatnot. So the pain aspect of inflammation is not attributed to an increased blood flow to the area where inflammation is occurring. So here's a summary of the innate defenses. Just make sure you go down through the physical and the chemical factors. Right here, I'll have a couple of questions on what they do. And over here, you can see the function dealing with each one of these and how they help eradicate pathogens from the body. For instance, I failed to mention vaginal secretions. Vaginal secretions are acidic, so it helps discourage the growth of bacteria. And the vaginal secretions help flush bacteria and other microbes out of the vagina to help eradicate the pathogens in that way. So each one of these has its own function to help get rid of pathogens out of our body. So just review that. Table 22.1. Summary of innate defenses continued with our second line defenses. These are pretty much our chemical defenses in our body. So make sure you review the chemicals, the various proteins. We're going to talk about the complement system in a minute. I haven't mentioned that, but just review this and know the functions of those particular innate defenses. And again, innate defense is a non-specific resistance to disease, which means our body's going to try and eradicate anything that looks bad, which is not normal in our body, in a non-specific manner. Unlike adaptive immunity. Adaptive immunity is referred to as specific resistance to disease. That means that when a pathogen enters our body, there will be a specific set of events, activation of cells, if you will, that will specifically destroy, hunt down, destroy those particular pathogens out of the body. That's why we call it specific resistance to disease. Specific resistance or adaptive immunity occurs in two main types. There is what we call cell-mediated immunity. I didn't write it all out, but cell-mediated immunity is abbreviated CMI, and antibody-mediated immunity, which is abbreviated AMI. So we're going to talk about all of the structures and cells involved with adaptive immunity. This particular diagram is basically a generic diagram showing both arms of the immune responses. So I'm going to go through this diagram with you and show you the outcomes at the bottom, and we'll get into details on some of this in a minute. But up here at the top, you see the thymus gland? The thymus gland basically is a big testing ground for immature T lymphocytes. So T lymphocytes are produced in red bone marrow, but they're not immunocompetent yet. The immature T lymphocytes are called pre-T cells. The pre-T cells enter circulation from the red bone marrow into the blood circulation, and they migrate to the thymus gland where they're tested. We're going to talk about the testing that goes on in a minute. But they're tested here, and they also mature here into different populations of T cells. So the pre-T cells are going to develop and mature in the thymus gland and become immunocompetent in the thymus gland. B lymphocytes are different. B lymphocytes, again, all blood cells produced in red bone marrow, including B lymphocytes and T lymphocytes, are produced in red bone marrow. The B lymphocytes are produced in red bone marrow, and they mature and become immunocompetent in red bone marrow, unlike T lymphocytes. So the thymus gland is only used to help mature, develop, and allow the pre-T cells to become immunocompetent. So once the pre-T cells become immunocompetent, they re-enter circulation and find their way to a home in what we call a secondary lymphatic organ or tissue. Remember, the secondary lymphatic organs and tissues include lymph nodes, the spleen, and lymphatic nodules. Secondary lymphatic tissues and organs are the areas where immune responses, adaptive immune responses, specific resistance to disease, see how many different names there are, occur. So adaptive immunity is going to occur or begin to occur in secondary lymphatic tissues and organs. So that's what this green little box represents, a lymph node or whatnot, a secondary lymphatic organ. So the mature T cells, when they mature out of a thymus gland, they develop into two main populations. Something called a CD8 cell, which develops into a cytotoxic killer T cell, and a CD4 cell, which develops into what we call a helper T cell. So when a CD8 cell is activated against a particular pathogen entering our body, it develops into a cytotoxic killer T cell. So the cytotoxic killer T cell is then further stimulated after it binds to a pathogen by cytokines or hormones that come from helper T cells. Helper T cells actually help initiate CMI initiated by the cytotoxic killer T cells, and it helps helper T cells help activate AMI, which is produced by activated B cells, which we call plasma cells. So CMI includes the ability of our CD8 cells to become activated against a particular pathogen into a cytotoxic killer T cell that then becomes co-stimulated from a helper T cell that will develop into two main populations of cells. Cells that are actively involved in killing off the pathogen, the invading pathogen, or what we call an antigen, and some of those cells stay behind as memory cells. And I'm going to talk about memory cells in a minute. The B cells, when they're activated against a particular pathogen, they develop into what we call a plasma cell. They also can be co-stimulated by a helper T cell, and in which case they too then develop into active cells called plasma cells that produce antibodies, and some of them stay behind as memory cells. The ability of our T and B lymphocytes to produce these memory cells is the reason why we get immunizations. Immunizations allow for our cells to become activated, but we retain the memory of that particular pathogen we were immunized against, so that our cells can later on in our life eradicate those pathogens much more easily before we get sick. So we're going to talk about these two arms of immunity here in a second. Just review the animation for adaptive immunity. You're going to have to look at this a couple of times before you get it down pat. So I want you to review all the material that you can in order to understand what happens during the adaptive immune response, or what we call specific resistance to disease. So before we talk about specific resistance to disease and the T cells and the B cells, let's talk about what antigens are. Yes, excuse me. So antigens and the receptors that bind them, referred to as antigen receptors, are going to be important in the process by which immune reactions occur. So we need to know a little bit about what antigens are. Number one, antigens are any type of molecule that can ultimately prompt an immune response to be activated in our body. Antigens that are not, let me word it this way. When a pathogen enters our body, we would refer to all of its molecules and the pathogen itself as a foreign antigen. So anything that is not us, that enters our body, not part of your own cells, is called a foreign antigen. But we also have antigens on the surface of our own cells, in which case we call those self-antigens. So our immune system cells have to be able to tell the difference between a self-antigen and a foreign antigen, so that our immune system cells basically does not wind up attacking our own cell. And we'll talk about that process in a second. But As far as an antigen is concerned, it has to have basically two different properties. Something called immunogenicity and reactivity. Immunogenicity is the ability of the antigen to prompt an immune response to occur in our body. Reactivity is the ability of the antigen to actually bind to an antigen receptor that is on one of our immune system cells, the T cells and the B cells. Ultimately, though, let's say a bacteria enters your body, the bacteria has hundreds of different types of molecules at its surface, and a handful of them may be reactive against a particular antigen receptor. So when we have what we call a whole antigen enter our body, there may be more than one reactive part of that antigen, smaller parts of it, in other words, and these smaller parts are called epitopes. Epitopes are, in this case, you see this might be a bacteria, we would call the whole thing an antigen. There are different regions or molecules at the surface of a bacteria that can ultimately bind to different types of receptors, in which case each one of these little molecules would be called an epitope, which can prompt an immune response, binding and then an immune response in our lymphocytes, T cells and B cells. In order for the immune response to be initiated, most antigens have to be processed first. So the processing of an antigen occurs in two main ways. Ultimately, the B cells or B lymphocytes have the ability to recognize the antigen itself in our fluids of our body, like lymph or our interstitial fluid, or in the plasma of our blood directly. So B cells can actually recognize an unprocessed antigen, and they themselves actually can become a processing cell for the antigen, which is called an antigen presenting cell. T lymphocytes, on the other hand, have to be presented with the antigen before it can recognize it, or fragments of the antigen before it can recognize it and thus mount a response against it. So in antigen processing, the proteins from the antigens are all broken down into smaller peptide fragments. And these protein or peptide fragments are ultimately bound to molecules at the surface of our cells referred to as major histocompatibility complexes or proteins, and for short we call them MHCs. So the MHCs come in two main classes. Ultimately, they have the job of demonstrating antigens at the surface of the cells where the MHC is found on. So the antigen MHC complex can be inserted into a membrane and then present the antigen to our lymphocytes. So how does this processing work? Well, as I mentioned earlier, there are two main ways that antigen processing occurs. There's something called exogenous antigen processing, and the only cells in our body that have the ability to process an antigen through an exogenous mechanism are cells that are phagocytes, which include macrophages. B lymphocytes themselves are processing exogenous antigens, and cells called dendritic cells. So how does the process work? Well, an antigen presenting cell, called an APC for short, first has to phagocytize the antigen. In this case, they show a bacteria. So they go through the process of phagocytosis. The phagosome, or what we call an endosome, is brought into the cell. It fuses with the membranes that contain the MHC molecules on them. In the case of APCs, the MHC molecule is referred to as an MHC2. The MHC2 molecule then binds to a fragment of the antigen that was phagocytosed into the cell. And the MHC2 protein, which is our own protein, actually presents the antigen at the surface of the cell. So this actually forms like what I like to call the red flag. So an APC, which includes macrophages, B lymphocytes, and dendritic cells, will always process endogenously process antigens through phagocytosis, digest the antigen down, and bind the parts of the antigen to the MHC2 protein. And the MHC2 protein then demonstrates the antigen at the surface of the cell, and the antigen presenting cell goes around our body and says, hey, this is what the bad guy looks like, telling that to our lymphocytes, B cells and T cells. So the T lymphocytes and the B lymphocytes can recognize this particular antigen. They become activated against it, and then they can go and do their job at eradicating that pathogen out of the body, which we still have to talk about. So, I'm sorry I said endogenous processing. I meant exogenous. Exogenous means outside of the body. So everything I said was correct except when I said endogenous. This is exogenous antigen processing. It's called exogenous because the antigen is on the outside of the cell. It has to be brought into the cell to be processed. So during exogenous antigen processing, we phagocytize the antigen, in this case bacteria, and present it at the surface of an APC on an MHC2 protein. So an antigen presenting cell processes the antigen exogenously and presents the antigen on an MHC2 at the surface of the cell. Sorry about that. During endogenous antigen processing, the actual antigen is processed on the inside of the cell. The antigen does not have to be phagocytized. So often these types of antigens that are being processed are from viral infections, like the COVID-19 virus that's going around. So a virus that infects our cell gets into the cell directly. So an antigen that gets into the cell directly without phagocytosis having to occur is called an endogenous antigen because it occurs within the cell. So the endogenous antigen ultimately fragments and becomes attached to another type of MHC protein called MHC1. Now, all of the cells in our body present MHC1 proteins at our surface, except for red blood cells. Red blood cells don't have MHC1s being demonstrated at the surface. The MHC1 proteins are always demonstrating antigens at the surface. Sometimes it's our own antigen. And if our own antigen is being demonstrated at the surface, our lymphocytes are not supposed to attack it. So if a lymphocyte attacks an antigen being presented by our MHC1 and the antigen is our own protein, then you develop an autoimmune disorder. That's exactly what we don't want to happen. So MHC1 proteins are always demonstrating endogenous antigens. In this case, it might be a viral particle from the viral infection that infected our cell and demonstrate it to a passerby or lymphocyte, can see it and become activated against it. But in this case, the cells that are infected with, say, the same virus are ultimately doomed to die because we can't save those cells. The cells in our body that are infected with a virus, that our lymphocytes can become activated against, the whole cell is going to die in our body. And that's our immune system's way of getting rid of the viral infection. So here you can see some different hormones, which are called cytokines, that ultimately are a part of our immune responses. I want you to review these, what are called interleukins, and what they do, the origins and their functions. I'm going to talk about a couple of them specifically in a second, but make sure you review this table to know what these particular signal molecules are doing. So let's talk about cell-mediated immunity. Cell-mediated immunity, first off, is carried out by a cell referred to as a CD8 lymphocyte, or a CD8 cell, or another name for it is a T8 cell. So the CD8 or T8 cells can become activated and proliferate, which means divide, into two main populations of cells. They can clone themselves into the effector cell, which is referred to as a cytotoxic killer T cell, and they can also develop a pool of cells that are identical to the cell that was activated against the pathogen in the first place. Those are called memory cells, and they kind of stay behind, so we always have a memory of the type of pathogen that entered our body so we can fight it off at a later time. So when the T8 cells are activated, they develop into a cytotoxic killer T cell, and then they go and eliminate the pathogen. So you can review this animation on cell-mediated immunity, and let's look at how the clonal selection occurs with, first of all, a helper T cell. So a helper T cell is one type of subpopulation of T cells, and the helper T cell's job is to help both cell-mediated immunity and antibody-mediated immunity occur in our body. I'll put it to you this way. If the helper T cell count in our body goes down, you become severely immunocompromised. This is exactly what happens with the HIV infection, which causes, as you know, AIDS. HIV infects a helper T cell and destroys them in the body, or the person that's infected with it. So when their helper T cell count goes way down because the virus is destroying those cells, they become immunocompromised, and then they die from secondary infections. Right? So let's see how we activate a helper T cell and how the helper T cell is cloned into its different populations. First of all, antigen-presenting cells process exogenous antigens. Those are antigens on the outside of the cell in our fluids in our body, or tissues in our body. So the antigen-presenting cell goes through exogenous processing and presents the antigen, which is a little purple diamond here, on an MHC2 protein. So if we look at this circle, it's enlarged over here. You can see the proteins and receptors that are involved with this activation process. So the MHC2 molecule on an APC has the job of demonstrating the antigen protein to the helper T cell. Now the T cells all have what we call TCRs. This stands for T cell receptor. And the TCRs, if they have the capability of binding to the antigen, they bind to it. So there's two binding events that are required in order for the activation of a lymphocyte to occur. There's the binding of the T cell receptor to the antigen, but there's also the binding of the CD4 receptor, which are located on helper T cells only. The CD4 receptor has the job of binding to an MHC2 protein. So the binding between the CD4 protein and the MHC2 protein on an antigen-presenting cell basically anchors these two cells together, which I like to call docking. The two cells dock together just like a boat docks at the dock. They bind together and they're held in place until the activation process can be initiated. So that's one binding event between the MHC2 protein and the CD4 receptor protein. Then there's a binding with the T cell receptor and the antigen. So that binding event occurs, it anchors the cells together, and then the activation process occurs. When the T cell is activated, the cell undergoes proliferation and differentiation, which is development. This is called clonal selection. So a population of T cells that were activated against this particular pathogen remain activated, and they start producing several different hormones, which are called cytokines generically, that help stimulate the cell-mediated immune response and help stimulate antibody-mediated immune response. So interleukin-2 is one of those principal hormones with a whole bunch of other cytokines that can be released by the activated helper T cells, and this helps activate both arms of the immune response. A subpopulation of the same exact T cell that was activated against this particular pathogen are not activated directly, but they stay as a long-term memory cell, so that if our body ever encounters the same exact pathogen again, we have a whole army of them that can be standing by and ready to help eradicate it out of our body. The first time we ever encounter a pathogen, we typically get sick from it because our immune response is slow and sluggish because we don't have memory cells yet. But every subsequent time we come in contact with the same exact pathogen, our immune system cells have a stronger and more rapid response to eradicate that pathogen because we have many more of those cells that can do so, and we typically don't get sick from it. It's not always the case, but the majority of the time, that is exactly what happens. So that was the helper T cell. What about the CD8 cell or the T8 cell and its development into a cytotoxic killer T cell? Well, very similar response here. Here we have an infected body cell. Maybe it's infected with the COVID-19 virus, maybe it's an influenza virus, whatever virus someone gets infected with, or an internal parasite. Those antigens are processed via endogenous processing, and that endogenous antigen then is demonstrated on an MHC1 protein. So let's look at this circle enlarged over here so we can see the proteins involved in the activation of a cytotoxic killer T cell. So here's our body cell that's infected with a pathogen. The endogenous antigen, which is a round purple dot, is bound to and demonstrated at the surface on an MHC1 protein. The T8 cells are called that because they produce and demonstrate CD8 proteins at their surface. So the CD8 protein on the CD8 cell or T8 cell has the job of binding to an MHC1 protein on the body cells in our body. So the CD8 receptor binds to an MHC1 protein and anchors the two cells together to give time for the T cell receptor to bind to the antigen. If the binding event occurs correctly, so then we can activate the T8 cell, but requires co-stimulation. So there's two binding events that have to take place to start the process. The binding of a CD8 to an MHC1 anchors the cells together, and then the TCR, T cell receptor, has to bind to the antigen presented by the MHC1. Those two binding events are required in order to activate the lymphocyte. But another requirement is co-stimulation. So the helper T cells that are activated against that particular antigen are releasing cytokines and hormones like interleukin-2. And interleukin-2 is the hormone that is going to co-stimulate the CD8 cell to help it become activated into a cytotoxic killer T cell. So once that co-stimulation with interleukin-2 occurs, we develop activated cytotoxic killer T cells that then go through the clonal selection of becoming activated killers, activated cytotoxic killer T cells that go around the body and attack and destroy all of our body cells that are infected with that same exact pathogen or antigen that it was activated against. So this infected body cell is doomed to die. Other populations of those same exact activated T cells can stay behind as memory cells, and that's part of why we have the ability to become immunized against pathogens because of memory cells. And so these memory cells stay behind for months and years at a time and allow us to have a faster response against that same exact pathogen if it ever comes into our body again, and they can eradicate it before we get sick. Now, how do activated cytotoxic killer T cells kill the infected body cells that they were activated against the antigen that they're infected with? Well, there's a couple of things that happen. Ultimately, the infected body cell has to die. So how does that happen? Well, activated cytotoxic killer T cells can release enzymes called granzymes. Now, granzymes ultimately, and of course, these cytotoxic killer T cells, I must say, roam around the body until they come in contact with an infected body cell and they bind to it in a similar fashion with the CD8 to the MHC1 and the TCR to the antigen. And when that happens, they release granzymes which poke holes into the infected body cell and causes the cell to undergo what we call apoptosis. Apoptosis is programmed cell death. They lyse open, and it releases all of the pathogens that can be phagocytized by other immune system cells in our body. Another way that the activated cytotoxic killer T cell destroys our cells is by releasing enzymes called granulysin and proteins called perforins. Perforins are proteins that poke holes or perforate the membrane in the infected cell, and granulysin actually gets into the cell and destroys the cell's proteins and DNA and causes the cell to die. So these are two main ways that activated cytotoxic killer T cells actually destroys our own infected cells in our body to eradicate the pathogen. Now, antibody-mediated immunity, or AMI, involves an antigen that is recognized and bound, first of all, and helper T cells bound by the B cell, and the helper T cells can co-stimulate the B cell so it can proliferate and differentiate into activated cells called plasma cells and also memory cells. So the activated B lymphocytes are called plasma cells. They produce antibodies that help eliminate the antigens out of the body. Now, antibodies don't kill or destroy anything directly. They just help eliminate it out the body. So B lymphocytes actually can be activated in two main ways. B lymphocytes themselves have the ability to bind to antigens that are not processed, in which case they can actually process an antigen and then present it to a helper T cell. But they can be activated without processing the antigen and or co-stimulation from a helper T cell, or they can be activated by processing the antigen and being co-stimulated by a helper T cell. So they can be activated in both ways. However, B cells become activated much more efficiently through co-stimulation from the helper T cell. So, like T lymphocytes have a T cell receptor that binds an antigen, B lymphocytes have a B cell receptor. So these B cell receptors bind to the antigen, they become activated, and the B lymphocytes form two populations of cells. Through clonal selection, they proliferate and differentiate into activated B cells called plasma cells that produce antibodies, and they develop a population of cells that stay behind as what we call the memory cells again, so that we have enough of them later on to eradicate the same pathogen more quickly if it ever enters our body a second, third, fourth time. Okay, so when B lymphocytes are activated, they're called plasma cells, and they produce antibodies. So antibodies are secreted proteins from the B lymphocyte, or I should say plasma cell at this point. So the antibodies have a particular structure. It's made up of heavy chains and light chains. The heavy chain is a long protein that we see here. You can see how it's shaped like a Y in this case. So we have what we call the heavy chains and then the light chains. But there's two main regions of an antibody that are of some importance. The main region that we talk about when binding to an antigen is up here, which is called the variable region. And you can see VL, that's for variable region on the light chain, and VH for variable region on the heavy chain. So there are variable regions on the antibody, and these are the regions of the protein that actually changes to allow for antibodies that are produced against pathogens to bind to a whole variety of different antigens. The rest of the antibody, and there's 5 major classes of them, I'll show you the names in a minute, the proteins themselves are all the same from this point down. So there's what we call the FC region, or here they call it the stem region. All of this is exactly the same on each class of antibody. The only thing that is different between the different types of antibodies that are produced by the plasma cells is the variable region. And that's due to the genes that are produced. The genes that are producing the protein are different inside the B lymphocyte. So the variable region, again, is a region where the antigen will be bound to. So here's a chart that shows the different 5 classes of antibodies. Just make sure you know a little bit about their characteristics and their functions here. IgG antibodies are what we call a monomer. They have to take the characteristic Y shape here. These are the only classes of antibodies that have the ability to cross the placenta. I think we talked about that in hemolytic disease of a newborn. So those anti-Rh antibodies were of the IgG class. But we also have IgA, IgM, IgD, and IgE. I'm going to have a few questions on antibodies, a couple maybe, on the test. So just make sure you review their particular roles in the body. All right? So let's talk about the complement system. The complement system actually, I should have put this in an earlier region of the packet, but because the complement system is part of our non-specific resistance to disease, is not what we call adaptive immunity, and the complement system is comprised of many different types of proteins and molecules that are flowing in the plasma of our blood. So I just want to go through a little bit about the complement system. There's a little bit more to it than what we see on this diagram, but each one of these represents a protein. So let's start at what we call C3. C3 is a protein that, when activated in our body, divides into 2 subunits. It's actually cut in half when it's activated. We get what's called a C3A and a C3B. The C3A activated complement protein helps increase the inflammatory response. In this case, they show the mast cell releases histamine, so C3A is aiding in this response, along with an activated C5 protein called C5A. So C3A and C5A, their job in our body is to help increase inflammation. And here they show just histamine being released, which is one of the inflammatory molecules. But C3b is of some other interest. So let's talk about that for a second. C3b is a activated C3 protein that goes and binds to the surface of an antigen. So all these little red bumps on the surface of this microbe over here are C3b proteins. So when C3b binds around the outside of a microbe like this, that's called opsonization. Opsonization, or coating the antigen, so to speak, helps our macrophages and other phagocytes find the microbe and phagocytize it. So opsonization actually increases the efficiency of phagocytosis and thus the eradication of the pathogen out of the body. But look what happens when we activate C5, and all of these proteins are activated in turn, by the way. C5, when activated, is also cut in half. We get a C5a involved in inflammation, and then we get C5b. C5b activates C6, another complement protein, and C7, which activates then C8. So all of these complement proteins are activated together to help form a complex which inserts in the membranes of pathogens. So this insertion of these proteins in the membrane allows for the last of the complement proteins of interest here, called C9, to insert in the membrane. They all start binding to the C5b, C6, C7, and C8 complex. And they bind and bind and bind, and they form a hole. This is called the MAC attack. The membrane attack complex forms a channel in the phospholipid bilayer of the cell, in this case, let's say a bacteria, which basically causes the cell to lyse open. So all of these activated complement proteins can induce phagocytosis, induce inflammation, and induce cytolysis of the cell through what's called the MAC attack complex, or the membrane attack complex. So it's called the MAC attack. So it causes the cell to lyse open when we poke holes in it. So that's one way we can get rid of the pathogens out of the body, through the complement activation system. All right, now let's talk about adaptive immunity with some definitions. I have a question on this or so. There's something called naturally acquired active immunity. Naturally acquired active immunity basically is when you come in contact with an antigen. You're exposed to someone that's sick and they give you the virus or the bacteria, and then your body naturally produces an immune response against it. The cell-mediated and antibody-mediated immune responses against that particular pathogen. So when you come in contact with that, that's what you get. The naturally acquired active immunity. Naturally acquired passive immunity is what happens when antibodies are delivered to us. Like a baby receives milk from the mother, receives antibodies across the placenta from the mother when developing in utero. So the baby is naturally acquiring the antibodies that the mother is making. And that's why it's called naturally acquired, but passive immunity, because the baby is not introduced to the pathogen from someone else. They're actually acquiring antibodies from the mother. Now, artificially acquired active immunity is basically what happens when you go get immunized, a vaccination. So when we get a vaccination, or we go get our immunization, we're basically artificially introducing the pathogen to our body. Except more often than not, the pathogen has been processed so that we don't get sick from it. So the processing of that pathogen in the vaccine allows our immune system cells to see the antigen, but the pathogen is not active in causing infection the majority of the time. So we become artificially acquired active immunity through immunization. Artificially acquired passive immunity is when we go to the hospital or the doctor because we have really bad infection and they hang a bag of immunoglobulins on us. So they actually can give us antibodies against a particular pathogen in our body to help eradicate the pathogen that way. It's called artificially acquired passive immunity. Now, the last couple things we have to talk about deals with adaptive immunity and really the development and maturation of T lymphocytes in our body. So, and B lymphocytes for that matter. Our lymphocytes have to be able to tell the difference between what is you from what is not you, or basically know what is a self-antigen relative to a foreign antigen. So we have what we call self-recognition, and that's where the T cells will be able to recognize that our cells are ours and won't attack it. And then something called self-tolerance. And self-tolerance is the ability for the T cell to not react to our own cells in our body. So we have what are called positive and negative selection pressures for self-recognition and self-tolerance. And here's the diagram for T cells that develop. And this process occurs, by the way, for T cells in the thymus gland. So let's just go through this together so I can show you what's going on with positive and negative selection pressures for the development of a immunocompetent T cell occurring in the thymus gland. So first of all, just read the questions. Does an immature T cell called a pre-T cell recognize a major histocompatibility protein? So they have to be able to recognize the major histocompatibility protein, or the MHC. Remember, the MHCs are the proteins that present antigens at the surface of all the cells in the body. So if the helper T cell or CD8 cell cannot recognize the MHC, then that T cell has to be destroyed. It has to die and not enter circulation, because it will not be able to do its job. So in order to recognize the MHC, and it can recognize the MHC, that's called a positive selection. If you recognize the MHC, you live. If you don't, you die. So if it does recognize the MHC, the next test involves the T cell receptor. Is a T cell receptor capable of binding to and recognizing our own antigens, what are called self-peptides or self-antigens? If the TCR recognizes, let's say yes, recognizes our own self-antigens, they have to die. We don't want the T cell being able to recognize our own self-antigen and binding to it and destroying our cells, so we need to kill it. So we do so. Those T cells are destroyed via what we call deletion, or cell death, or apoptosis, and something called anergy. The cell still lives, but it's inactivated. It's inactivated. So if we have a positive selection and can recognize the MHC, that's good. If we have our TCR recognize our own antigen, that's bad. So this is what we call a negative selection. So if the TCR is incapable of binding to our own self-antigens, which in this case would be no, those are the T cells that live. And only about 3% of all of the pre-T cells that are undergoing this testing in the thymus gland actually re-enter circulation to help become part of what we call cell-mediated immunity. All right? So what happens? A mature T cell in our lymphatic tissues becomes activated. Well, if an antigen recognition is accompanied with co-stimulation, that is to say, the T cell is activated and is co-stimulated by a helper T cell, then we have that particular T cell proliferate and differentiate into cytotoxic killer T cells and the memory cells that I was referring to before. If we have a T cell that is recognizing an antigen but without co-stimulation, we want that cell to die. This is a protective mechanism to help us prevent autoimmune responses from occurring. If we don't get co-stimulation, we want the cell to become inactivated, or we want deletion to occur, or the cell to die. So this is what really we call the selection process during, at least at the top, during development of the T cell in the thymus gland, and the selection process of the T cells that survived from the thymus gland, what happens to them in the lymphatic tissues in the body. So make sure you review this chart just to know what happens during the positive and negative selection process, and what happens out in the secondary lymphatic tissues if we get co-stimulation or if we don't get co-stimulation. Okay, so we also have B cell development and recognition and self-tolerance, self-recognition and self-tolerance that has to occur. First of all, does the immature B cell that's in the red bone marrow recognize our own MHC molecules? In this case, it's different. If our B lymphocytes does not recognize our own MHC molecules, which is called a negative selection. Basically, if you can't do something, you win. Negative selection, those B cells will move on. If we have B cells in the red bone marrow that can recognize and bind to our own MHC molecules, we want those to die. So they go through deletion and cell death in the red bone marrow and will never enter circulation. So the first step here for self-recognition is a negative selection pressure. So if they can't recognize MHC molecules in our own cells, that's a good thing for B cells. It's just the opposite for T lymphocytes. Make sure you review that. So the mature B cell then enters circulation, and if they have the ability to recognize an antigen, which is called the first signal, and they get co-stimulation, they then become activated and proliferate and differentiate into a clone of plasma cells and memory cells. In most conditions, in most cases, when a B cell recognizes an antigen but there's no co-stimulation, they undergo inactivation so that we don't have an overactive immune response in a situation where there is no foreign antigen. If the B cell has actually been activated against one of our own antigens, there'll be no co-stimulation. So those B cells will then be either destroyed or they will become inactivated through a process called anergy. Now the last chart that I included in the packet discusses the antigen presenting cells I mentioned earlier, macrophages, dendritic cells, and the B lymphocytes and their functions. So I want you to review that. And then the various types of lymphocytes and their roles in immune responses. What are cytotoxic killer T cells? What do they do? How do they do it? What are helper T cells? What is their role? What are the memory cells, T cells and B cells, and their roles? And so just review all of these different cells and what they do from this table. Might be good to do that beforehand, but just go back and review it when you're done with the PowerPoint. All right, so if you have any questions, I know this was a fairly long video. The chapter's not that easy. I want you to review the video and the PowerPoint several times. Our information that's included in here is supplemented with a note packet that I put in Canvas. So make sure you review the note packet for the lymphatic system as well as the PowerPoint. And if you have any questions, just email me and I'll be glad to answer them. All right, so that's the end of the video. Just email me if you need me.